So is our second chapter is reproduction in flowering plants. In this chapter, we have to see various type of reproduction that occurs or the events in sexual reproduction in flowering plant. So in sexual reproduction in flowering plant, it comprises of three events. First of all, pre-fertilization, then fertilization and post-fertilization events. So for that, we should know that what are the main structures that will take part in this. So flower structure we have to see, like first we will see pistil, stigma, style and ovary, ovule, sepal. The stamens are anther, pistil, filament. Pre-fertilization events is different for both male and female. In male, stamen and microsporangium and pollen grain formation take place, whereas in female, pistil, megasporangium formation and embryo sac formation take place. This is their diagram. Then it comes, what is pollination? Pollination we have to see, and its type. Pollination is autogamy, geitonogamy, or xenogamy, where agents are biotic or abiotic. That will be, we can say, wind or water, or even animals. Some example, like winter pollinated example is grass, water pollinated is Vallisneria, insect pollinated is yucca. Outbreeding devices, that are devices which promote cross-pollination. These are pollen release and stigma respectively not synchronized, and some other like self-incompatibility, arrangement of anther and stigma at different position, production of unisexual flowers, then pollen release and stigma respectively not synchronized. Then pollen pistil interaction, it is the interaction between pollen which comes from male and the or the pistil of female. So at this, it is a dynamic process involving pollen recognition followed by pollen tube growth. Actually, what happens when the pollen comes, it lands, and then it is being recognized by the some enzymes, we can say, or receptors present here. Then a pollen tube germinates. So this full event is shown here. Then artificial hybridization, it is one of the major approach of crop improvement using desired pollen grain for pollination, which can be achieved by emasculation and bagging. Okay. Then double fertilization is the important event. Double fertilization can be simply said as syngamy plus triple fusion. Syngamy is the process, and triple fusion, when we combine these two, then it can be told, or we can say it as a double fertilization. Then post-fertilization event comprises of development of seeds. Development of seed, then seeds can be two type, albuminous and non-albuminous. The endospermic seeds are known as albuminous seeds, whereas non-endospermic seeds are known as non-albuminous seeds. Then development of embryo. On this, embryo development can be of two type, one in dicot and second in monocot. Then development of endosperm also take place. Then formation of fruit can be further divided into two types, true fruit and false fruit. When parts other than ovary contribute to fruit formation, we say it as a false fruit. But when the fruit develops from ovary, we say it as a true fruit. Example is mango. Then some special modes are there, like parthenocarpy. It is a development of fruit without fertilization. And polyembryony, it is the occurrence of more than one embryo in a seed. Actually, there is only one embryo in seed, but if it happens then more than one, then we say it as polyembryony. Then apomixis, and significance of seed dispersal and fruit formation. Seed dispersal help the species to colonize in other areas, and many fruits have mechanism for dispersal of seeds. So this was our chapter in basic. Now we will just go through it. Sexual reproduction, it is the process of development of new organism from two parents. This is the diagrammatic representation of various parts of flowers. Then pre-fertilization events. Pre-fertilization events can be studied under 4 points. That is pollen grain germination, embryo sac formation, pollination, pollen pistil interaction. Then pollen grain formation, it is a male reproductive. We know that pollen will be formed from male. So male reproductive unit is stamen. Then development of anther. This is the diagram or the structure of anther. Walls of anther are epidermis, endothecum, middle layer, tapetum. Tapetum is the innermost layer of anther wall surrounding the sporogenous tissues. This is the nutritive layer. This is the structure of anther. This is the transverse section of anther. Then microsporogenesis is the process of formation of male spore. Micro is referred to as male. This potential pollen, or we can say the pollen which is potential to fertilize further is there, and the cells of sporogenous tissues forms microspore tetrads after meiosis, and that comprises of 4 cells haploid which stay together, and then pollen grains are formed. While the anther matures and dehydration occurs and dissociate, tetrad cell separates. These are different shapes. Microspores are separated from the tetrahedral arrangement. Then this is the basic general information about pollen grain. Pollen grain varies in shape or etc. This is then development of male gametophyte. These are the pollen products, that are the products formed after the pollen grain or we get from pollen. Then pollen viability, that means how we can store pollen or for at which temperature it is being stored. Then some pollens causes allergies, some type of allergy, or it is a kind of disorders like asthma and bronchitis. So this is known as pollen allergy. Then female reproductive unit is pistil. There's some basic information is given. It is the forms of ovule. This is orthotropous ovule, types of ovule, or we can say this. The body of ovule lies straight and direct over the funicle. This is the funicle. What it means is, if I make this as my funicle, the body lies in this way. The body lies in this way. So this we state or we say it as orthotropous. This is anatropous. This is some basic features. This is hemianatropous, camphytropous, amphitropous. Then megasporogenesis, formation of mother cell. Megaspore mother cell, which is a diploid cell near the micropylar region, which is present after meiosis, form megaspore haploid monosporic development occurs and forms megaspore mother cell, which is haploid. Then functional megaspore to embryo sac. Then this further forms embryo sac. Then formation of embryo sac. This is the diagrammatic representation of formation of embryo sac. And pollination. Pollination is the process in which the transfer of pollen grains from anther and to that of stigma of flower occurs. Pollination is of two types, self-pollination and cross-pollination. This is some examples and types of pollination. Pollination is of two types, as I told, self-pollination and cross-pollination. And that means self-pollination, same flower, different flower of same. When we say this, that if I say the pollination is happening, suppose I make this one plant, here one flower, here one flower, this flower is bisexual. This flower is bisexual and the pollination happens within this flower, then we state it as autogamy. Whereas if I make this plant, this one flower, and this as second flower, and if the pollination occurs within these two flowers, then we refer it as geitonogamy. That means different flower of same plant, and this is pollination in same flower, this one flower. Then chesmogamy and clastogamy. Then cross pollination is two flowers of different plants, easy to understand. Then what is the significance of pollination and some events that will happen after the pollination? After the pollination has happened, this will happen. Then it is double fertilization. Double fertilization can be termed as syngamy and triple fusion. This is the diagram. Then post-fertilization, that means fertilization is over. Now post-fertilization structure and their events. The first event is formation of endosperm. So basic knowledge is given. Then endosperm type is given. Function of endosperms are, they are given. Embryogeny is given in monopod. And so these are some structure that we have to learn and see, which are similar. Because if I say ovary, if I say ovary converts into fruit after fertilization. Ovary wall into pericarp, ovule into seed, integuments into seed coats, outer integument into testa, inner integument into tegmen, micropyle to micropyle remains same, funicle stalk of seed, nucellus, perisperm, egg cell, zygote, synergid, disintegrate and disappear. Formation of seed and fruit is shown. Fruit is also known as ripened ovary or fertilized ovary. These are some structures similar to post-fertilization. These are type of seed, exalbuminous and albuminous. Now, importance of seed. Why seeds are important? Then what is the viability? Then it comes apomixis, that some special cases, as it was told. Apomixis is a Greek word comprising of two words inside it. Apo, without, and mixis, marriage. That means without marriage, without fertilization, the formation occurs. So the organism reproducing through apomixis is called apomix. Then parthenogenesis, which is individual from a single gamete without fusion with another gamete. Then polyembryony. Parthenocarpy is formation of fruit without an event of fertilization. This true fruit, false fruit, and parthenocarpic fruit. In parthenocarpic fruit, no seeds are visible. Importance of parthenocarpic fruit is they do not contain seeds which have to be removed before eating fruits, so we can directly consume it. Fruit can be developed into the greenhouse where pollinators are not available, and quicker food processing. This is the flowchart of this chapter. These are some special questions which are asked previously. So this was our chapter number 2.